Okay, so this is part 2 of chapter 26, and we're going to talk about filtration. And so remember there was filtration, reabsorption, secretion. We're mainly going to get started on filtration, and then we'll talk about reabsorption and get into the countercurrent mechanism, and that's all we're going to cover for this part. So that's actually in the slides that go through chapter 26. Okay, and also GFR, glomerular filtration rate. All right, so first of all, the last thing we were talking about was urine production and how urine production takes place. We looked at the big anatomical parts of the kidney, and then we got down to the nephron, which this is a nephron right here. And we talked about the glomerulus, which is the capillary ball, the Bowman's capsule, which surrounds it. Those two things together make up the renal corpuscle. So as filtration occurs, fluid without blood cells and large proteins, fluid, which is essentially blood plasma, moves into the proximal convoluted tubule, which is this part, and then you have the descending loop of Henle, the ascending loop of Henle, the distal convoluted tubule, and then the collecting duct. Okay, so, and these are the processes that take place: filtration, reabsorption, secretion, and then whatever's left that moves out is called excretion. So that's what's excreted. So what we have here is a picture of a nephron. So essentially what we've done is we've taken this and we've straightened it out. And so we can see that as blood moves in through the afferent, so as blood moves in through the afferent arteriole, and then you have some of that fluid that leaks out. That's the filtration that takes place. And then it leaves through the efferent arteriole. Okay? Um, so you can see that the fluid that makes it through, some of it is going to be reabsorbed. So the fluid and ions and some other things that we'll talk about are going to move out of the tubule and back into blood flow. Okay, so that's reabsorption. And then we also see that some things that just happen to make it by this can actually have a second chance of moving into the tubule, and that's called secretion. Okay, so this is reabsorption when it moves out of the tubule into the blood. Secretion is when it moves from the blood back into the tubule, and then whatever's left becomes urine. Okay, so cleaning your blood starts with filtration. So that's the renal corpuscle that I mentioned earlier. The renal corpuscle consists of the Bowman's capsule, which is this area around, okay, which is actually part of the tubule. Okay, so that's the Bowman's capsule, and then the glomerulus, which is the capillary ball that sits right here. Okay, so blood enters, as I said, blood enters through the afferent arteriole, and then filtration occurs, and then any blood that's still left, which is about 80%, will flow back out. Okay, so about 20 to 25% of cardiac output goes to the kidneys, mentioned that before, and about 20% of that blood is actually filtered into nephrons. So that means it actually leaks through that glomerulus and moves into the tubule. Okay, and then of course, since that's quite a bit of blood, that's going to, or it's fluid at this point because it doesn't have blood cells anymore, most of that is going to be reabsorbed. Okay, so how does filtration work? There are a couple of things. So if we really look at this in a lot more detail, we can see that here's our afferent arteriole moving in here, and then you can see that there are these specialized cells that sit around it. And what these cells do is they cause a filtration mesh to sort of form over it. And so we have these little holes here. These are called fenestrae. And these little holes will let fluid go through it. Now you can see here are some blood cells here. And you can see that those are way too big to move through. So those aren't going to be able to move through, but the fluid can, and little tiny things like ions can, and maybe some really tiny proteins, and glucose. Glucose can move in there. So a lot of small things can move through there, but large things like these larger proteins like albumin can't move through, and so those stay in the arteriole. So they stay in the capillary that's here, and then they remain in the arteriole, and they move on. Okay, so these specialized cells are called podocytes, cell processes laid down by the Bowman's capsule. Okay, they look like little feet, and so that's why they're called podocyte. Like a podiatrist is a foot doctor. These are podocytes, so foot cells. And then the fenestrae, small pores in the glomerulus capillaries, which is what we talked about, which I showed you right here, which are very, very tiny. So that's why blood cells and proteins aren't supposed to be able to fit through. And if you do have blood cells, if you do see blood in your urine, that's usually not a good sign, because that means that this hole has been breached and blood cells are starting to move through. Okay, so we can measure something called glomerular filtration rate. And so kind of what I just said about what happens if this filter, this glomerulus, becomes leaky and those holes become too big, that's going to cause the filtration rate, the rate at which fluid moves through there, to become a lot quicker. Okay, so the filtration that takes place, we call it, I mean, once it's filtered through, it actually goes into the nephron, we call that filtrate. And so that's going to have water, small molecules, like I said, like glucose and sodium and potassium and calcium. Small molecules, ions that can pass through into the nephron tubule. And so this glomerular filtration rate is the amount of filtrate that's produced each minute by your kidneys. And usually it's around 180 liters per day. Okay, so that's a lot. Okay, since really our blood volume is only about 5 liters, that means we're cycling through the entire, all of our blood several times, many times during the day. Okay, so the average urine produced in a day is only 1 to 2 liter, which means we have to kind of replenish that with water. And so that means that since there's 180 liters per day being filtered, that means that most of it has to be reabsorbed. Okay, so that's what we're going to start talking about now. Or after we get done talking about glomerular filtration rate. Okay, so what keeps fluid flowing? Okay, so we've talked about this before, and you can see several different arrows. If you look here, you can see several different arrows. A couple of them are pointing back in, and then there's a big arrow, okay, pointing out. Okay, out in, depends on your perspective, but it's coming from this capillary and moving into the nephron. You could see that this is capillary pressure, or what we called hydrostatic pressure when we were talking about this in the cardiovascular section. So that means blood pressure. So the blood is pushing out, but you also have osmotic pressure. Okay, so that's what we call blood colloidal osmotic pressure. Sometimes it's called oncotic pressure. But that's because once this fluid starts to move out, you're leaving a bunch of cells and proteins, and there are other molecules in there that are kind of, remember with osmosis, where you have lots of particles, and if you don't have very many over here, well, water is going to tend to move in this direction. Okay, so that's what's going on. Water is kind of pushing back a little bit. Okay, and then you have the whole issue of pressure building up in here, and that's also pushing back a little bit. But overall, when the whole thing is said and done, the blood pressure, okay, so this is called glomerular capillary pressure. Well, capillary pressure is that blood pressure, and that's going to win. And so you're going to have net flow into the nephron here. Okay, so GFR is usually very constant, and there's a good reason for that. So first of all, why does it matter? Why are we even talking about GFR? Well, as I mentioned, if you have low GFR, so if your glomerular filtration rate is very low, then that might mean that something is blocked. So something in your glomerulus up here, those little fenestrae and the podocytes, those things that were making the mesh, making that filter, are somehow blocked. It could be, from a bacterial infection or something like that. It could be an immune response where immune cells are building up in there. Somehow it's getting blocked. It could be damaged and replaced by scar tissue. Lots of different things could be causing that to be blocked. Or it could be broken down. And that means that the glomerulus is just leaky. The holes have gotten larger. There could be damage to that capillary. And so that's going to let things move through more quickly. So if you think of this as a filter, okay, so if I draw this as a filter and fluid is trying to move through, okay, so it's going to have to go through those little holes, okay, and then the blood cells are going to be left behind. This isn't a very good picture. if I make those holes really, really big, first of all, the blood cells could get through, but you're also going to have much higher flow through. And that's what GFR is. It's the rate at which things move through that filter, that glomerulus filter. Okay, so how do we measure it? Well, our muscles are constantly breaking down a um, a molecule called creatine. Okay, so the muscles are constantly working, and usually at a pretty constant rate, unless you're doing a whole lot of exercising or just doing something physically that's out of the ordinary. Usually we make this molecule called creatinine. So our muscles make this molecule called creatinine. And creatinine is just a breakdown product of something else called creatine phosphate. I don't know if you remember back from the cardiovascular system, but creatine phosphate was just this little thing that held on to a phosphate group, and then it would transfer it to an ADP if you needed a very, very quick source of ATP. Okay, so this is creatine. This little molecule here was creatine. And well, that gets old and it breaks down and it turns into creatinine, and it's constantly being released into the blood. So we can see by measuring how much of this is in your urine and how much you release over a certain amount of time, we can see what your GFR is. And that can give us a pretty good idea. So we can use a urine test to actually measure what your glomerular filtration rate is. So if you have pain in that area or something else is kind of cueing you off, then a urine test could actually say, okay, well, your glomerular filtration rate is reduced, and then you can look for further problems to see what might be causing that to take place. Okay, so how do we, on a day-by-day basis, control the glomerular filtration rate? I mean, what happens when you start running or exercising or something like that? Your blood pressure will increase. So does that just mean that you're just pushing a ton of fluid out through this filter, through this glomerular filter? No, it doesn't. We can actually control how quickly things move through. Therefore, we can control, well, that's controlling the GFR, the glomerular filtration rate. Okay, so if you look at the afferent arteriole and the efferent arteriole, you can see around it these muscle cells. Okay, so that's what these guys are that are surrounding it. These are muscle cells. And these muscle cells can contract or they can relax. And so they're going to make the opening going in either small when they're contract or large. Okay, and we already know that if it constricts, that you're not going to have as much blood flow. So that means you're reducing the blood flow through this afferent arteriole. Therefore, you're reducing the blood flow into the glomerulus. Therefore, if you constrict it, you're slow. I don't know. I'm trying to be cute by doing that, but it just looks like slewing. You're slowing your GFR. Okay, by constricting this, because you're reducing the amount of blood flow. So that means when we start running or exercising and our blood pressure increases, then all your body has to do is kind of clamp that shut a little bit, and your GFR remains constant. And that's what this graph down here is showing. It's showing that over a range of blood pressure, so this is mean arterial pressure, so it's the average approximately of your blood pressure, between 80 and 180, we could keep that pretty constant. Okay, all right. So, and that's how you do it. You can constrict the afferent arteriole and let less blood flow through, or you can actually, if you constrict the efferent arteriole, think about what happens there. If you block this off and blood's still moving in, you're actually increasing pressure, and so more fluid will go through, and that means you're increasing GFR. So it kind of makes sense without memorizing. If you have to memorize it, you can. But relaxing the afferent arteriole will increase GFR. Contracting it, it's going to restrict blood flow, so it's going to decrease your GFR, and that's what I drew here. Actually, I can erase that, so because sometimes you increase it. So and then the efferent is actually going to just have the opposite effect. If I constrict it, well, blood that's trying to flow through this and then it's trying to escape through the efferent, well, if I've constricted that, that blood's going to end up staying in here, which is going to increase the GFR. Okay, And that's what this says. So remember that it's on both. It's on both the afferent and the efferent. But that's what this says here. GFR is adjusted by controlling the rate at which blood flows into the nephron by contracting or dilating the smooth muscle in the afferent or the efferent arterioles. I have it in parentheses there, and just to anyways, but I circled it. It's not in parentheses because it's not important. It's I don't know why it's in parentheses. Okay, but remember that. Okay, so after filtration, things move into the renal tubule, and so we've got filtration that happened in our glomerulus here. We looked at the podocytes, all that stuff. Well, now the fluid, the filtrate that moves in first of all to the proximal tubule, or the proximal convoluted tubule. Sometimes we abbreviate that as PCT, proximal convoluted tubule. So what's in it? So what's left? We know that blood cells were left out. We know that large proteins like albumin, which is a large protein that's in the blood, those things didn't make it. So those things stayed in the blood. They just went right on through. But what's in there? So water is in there. Obviously, we know that water's there. Glucose is small enough to fit through there. Urea, which is a breakdown product of proteins. We're constantly breaking down proteins. Okay, and we form something called urea, which they've named it after the urine because that's where it's found, is in the urine. Ions are in there: sodium, chloride, potassium. Amino acids. Okay, it's you have amino acids through your body, whether they come from breaking down proteins or they just haven't been built up into proteins. But there are amino acids in your blood, and those can move through there. And there are lots of different waste molecules. I'm not going to go through any because right at the moment I can't think of any, but there are lots of waste molecules that are formed. Acetate is one. Okay, so there's one. All right, so no red blood cells or large proteins should be present. I think I've said that enough times. Probably because this is a clinical symptom. Okay, proteinuria. That means that there's protein in your urine. Okay, and that's a bad thing. All right, so once something, or it tells you something is going on, and it's usually bad. So once something moves into the tubule, its next stop is excretion. Okay, so that means that it's flowing through, and so the only way that you're going to keep all of that water and all of those ions is if you decide to reabsorb them, which is what you're going to do. Okay, so now we have to really understand what's going on with reabsorption. So now we've kind of said that we don't want to, that we've got all of this fluid moving into our kidneys, into our nephrons all the time, but we don't want it to turn into urine. So we know that with this reabsorption has to take place. And it's really interesting. And we can kind of remember this as a process. And I put numbers here. You know, a lot of this stuff all happens kind of at the same time. It's all happening constantly. But if we look at it in an orderly way, we can say that sodium is actively pumped out. So we have the sodium-potassium pump. Okay, so remember, we've been talking about the sodium-potassium pump since the very beginning. And what it does is it pumps sodium out of the cell. And that's what it does here too. It's pumping sodium out of the cell. Okay. Well, that's causing a concentration gradient. And so here you can see in this first one, so this is the tubule here. Okay, so this is your nephron tubule, and this is, I guess, extracellular fluid, and then it's going to move into a capillary eventually. But you can see that we're pumping the sodium out, we're keeping the sodium out of the cell, which means all you have to do is put a channel in there and say, hey, sodium, do you want to go into the cell? And the sodium says yes, and it says okay, and you're taking chloride with it. So that means that chloride, and that's what I have down here is number 2, chloride always follows sodium. So there goes the chloride. Okay, another thing that happens is sodium, hey, do you want to leave the cell? Yeah, but you have to take glucose with you. Hey, sodium, do you want to go into the cell? It says yes, but you have to take these amino acids with you. Well, we've got sodium leaving, we've got chloride leaving, we've got glucose leaving, amino acids are leaving. There are other ions that are leaving. All you have to do is open a channel for them. Everything seems to want to take off. And so what does that do with water? So now all of a sudden our ions are leaving, okay? And we're left with a whole lot of water. So what does water do? It's going to follow, because that's what water does. When you have lots of particles on one side and they're all leaving, then the water is going to also leave. Okay? So this all started with the sodium-potassium pump. So sodium is actively pumped out, chloride follows sodium, water moves out by osmosis because solutes are leaving, and then there are these other ones, the glucose, the urea, the amino acids. Those are also always kind of leaving. Okay, so this is happening at the same time, but it's really triggered by this active pumping out of sodium. Okay, so simply put, sodium gets actively pumped out first, then everything else passively follows. Okay, chloride, water, glucose, amino acids. I have some things named there. All right, so I mentioned glucose, and glucose is really important, especially because of the clinical value, I guess, of diabetes. But if we think about it, what are the first things that we recognize in someone who hasn't yet been diagnosed with diabetes, but what are the first symptoms that we see in someone who, and I'm talking mostly about type 2, but also type 1. The first things we recognize are frequent urination. Okay, so I'm just gonna put this down here, or circle it down here. A higher urination volume. So frequent urination is one of the first things we see with someone who is newly diagnosed with diabetes. The other thing we're going to see is thirst. Okay, and those are related, of course. If you're urinating, if all your fluid is leaving through your urine, well, you're going to be thirsty. Okay, and then of course, then we measure the urine, and what do we find? We find sugar. I'm going to cross that out and write glucose, because that's actually what we're measuring in the urine. Okay, so that's what we see. So what explains this? Well, what explains it is that if you have a hard time controlling your glucose levels in your blood, so your blood glucose levels get really, really high. Okay, now that's a problem of diabetes. We're not talking about what's causing diabetes, but the result is your blood glucose levels get really, really high. And so you have these things called glucose transporters. Now, one thing we have to remember is that your body likes glucose. It will absorb it all in the gut, so anything you eat that has glucose or sugar in it, your body's going to absorb it. Not only is it going to absorb it, it's going to keep it. So all of that glucose that's moving into this nephron tubule is going to be reabsorbed. Your body's not going to waste it. It's going to bring it right back through and then put it right back in. I'm going to draw a capillary over here. It's going to put it right back into your bloodstream. Okay, that's what's going to happen every time unless you have too much. Okay, because these glucose transporters, there are only so many of them, and when you have, look at this, it's like an army of glucose coming in there. There's no way these guys are going to be able to handle that when your blood glucose levels get too high. Okay, so what happens is they become saturated, and then you end up keeping the glucose in your nephron, okay, in that nephron tubule. So even though you're trying to get rid of it, if it's too high, it stays in there. So think about what I was talking about with osmosis. So if this is the inside of the tubule and this is all the glucose, okay, when we're trying to make, when we have these particles that are leaving, well, the water's supposed to follow those, right? Well, it is supposed to follow it, except it says, wait a minute, there's a whole lot of glucose in here. Maybe I don't want to leave. Maybe I don't want to leave the nephron, and I'm just going to go ahead and stay right in here. Some might leave. Okay, well, it has to, because otherwise you would die of dehydration in a few minutes, but some of it will stay in. So you end up with more water. Okay, more water stays in the nephron. And so if you have more water in the nephron, guess what? It's going to move all the way through, and it's going to leave as urine. Okay, so that's why you urinate more when you have too much glucose, because your transporters get clogged up. Okay, they get backed up. You can't get it all. You can't get it all out, and so that glucose stays in there, which is going to mess with the osmotic potential, and that water is going to tend to stay in the nephron and then you're going to have more urine, and then the thirst is easy. You're urinating out all your water, okay? And so you become thirsty. All right. So let's see. So these things that I was talking about, I'm gonna switch back to this. When we were talking about this, we were talking mainly about the proximal convoluted tubule. Okay, so we were talking primarily about this part. Okay. Now, so that means that about 80% of reabsorption happens here in the proximal tubule. So that means most of the water, most of the salts, most of the glucose are reabsorbed just as soon as they make it in there. 80% of it gone. 20% still quite a bit though. Okay, so then what happens is you move from the proximal tubule down into the descending loop of Henle. Okay, descending loop of Henle. But now things start to get different. Whereas in the proximal tubule, everything was leaving. Salts were leaving, glucose was leaving, water was leaving. Everything was being reabsorbed. Here, it's only permeable to water. Okay, so only water. I should probably make that blue. Okay, so only water is moving out and being reabsorbed in the descending loop of Henle. Okay. And the reason it has that, the water is able to move out is because well, first of all, we don't have any channels for the ions, so the ions can't move through, but we do have these little channels called aquaporins that let the water move out. Okay, so water is just rushing out as much as it can. Now, one thing we have to remember is that at the base, it's very concentrated. But when I say the base, I mean the medulla. Okay. So this is in the medulla, deep down in the kidney. It's very concentrated. But here at the cortex, which is the outer part, it's very dilute. Okay? So that means as the fluid is moving down, we have these little pores that let water move through, and the water says, wow, look how concentrated it is out here. Look at all these particles out here, all these solutes out here. And so, of course, that water is just going to keep moving out. Almost squeezes the water out of it like squeezing a rag or something. So the water moves out. And so now we've gotten rid of even more water. Now, so that's what happens in the descending loop. Now, in the ascending loop of Henle, it's only permeable to solutes. Okay, so what's moving out of here? Sodium, potassium, chloride, that's moving out. Okay, and that's going to be moving down its concentration gradient. So right here, maybe because it's so concentrated, not as much moves out, but then as you move up, more and more particles are going to move out because now it's very dilute up here. So it's very dilute out in the cortex, and so these particles start to move out. Okay, so that's what's happening. That's what's happening in the loop of Henle, and that's what you have to remember. You have to remember that descending loop is only permeable to water, and that ascending loop is only permeable to what we usually call salts. Okay, and primarily we're talking about the sodium, potassium, NKC, chloride. Okay, so that's mainly what we're seeing there. All right. So this is kind of saying the same thing, except now we have numbers, and these numbers can be very, very helpful without even knowing what this is. Okay, it's milliosmoles. Sometimes we say milliosmoles per liter, which is milliosmolar, but whatever. All this is doing is it's giving you a nice simple way of saying, gee, how dilute or how concentrated is this? A small number means it's dilute, and a large number means it's very concentrated, okay, very thick. Okay. And that's what we see down deep in here. So that again explains why the water is able to move out. Okay? And as it moves out, it moves into, or it has the ability to move into what's called the vasa recta, or a capillary, or a series of capillaries that are outside it. Okay? Now, when we were talking earlier, and when we talk about this water, this is kind of an important idea to remember, is that we said the water moved out over there, and then the salts moved out over here. Okay? But we also said that the concentration is higher down here deep in the medulla and very dilute up here on top. But if you look at this, it almost looks like, well, the water's coming out over here, so it should be very dilute on the left side. Okay, so it should be very dilute over here and very concentrated over here on the right side. Okay, but that's not the way it is. And the reason for that is because of something called the countercurrent mechanism. So that means that as these salts are released here, they move right into the blood, okay? Because the blood's only about 300, and then these are a lot more concentrated down here. And so those salts are constantly moving into this vasa recta, this capillary. And this capillary is going in the opposite direction. So it's pulling them down, okay? So it's pulling these salts down. Now the same thing is happening over here. So that means that this concentration that was supposed to be over here on the right is actually being pulled down, okay, constantly all the time. Now the water, what about the water that was building up over here? Well, it's the same thing, that as the water is being released, it is constantly being pulled up. Okay, so the water is being pulled up to the top all the time, okay, which is going to make the top or the cortex very dilute. Okay, and that's this down here is very concentrated. Okay, so we call that the countercurrent mechanism. Okay, this term right here. Okay, so the fact is that the cortex of the kidney is very dilute, the medulla of the kidney is very concentrated, and how is that? It's because of the countercurrent mechanism. So that means as this water is moving out, it's being swept up and moved to the top or the cortex. As these salts are moving out, they're being swept down and moving them down to the bottom. Okay. So hopefully that makes sense, and your slides will say the same thing on that. Okay, so that's pretty much it. We have, or not, it's not really pretty much it. That's it for this recording. But I do have this here that's kind of trying to keep everything in perspective. So as things move through the filtrate, as things are filtered in the glomerulus, 100% is produced. Okay, so that just means that everything that moves through the filter moves into the nephron. 80%, as I mentioned before, of filtrate is reabsorbed right here in the proximal tubule. Remember that. Remember that that's where it takes place. I mean, that's an easy thing to remember. It's an easy question if you ever have it on a test to remember. The loop of Henle, about 6%. You don't have to remember these numbers here, but you can see that primarily it's at the proximal tubule, and then it kind of drops from there. But ultimately, what we're looking at is trying to get 99% absorbed. Okay, so we'll continue with the rest of it in the next video.